Okay, so welcome everyone to the STEM talk of the sixth Polygent Symposium of Rising Scholars. My name is Alyssa Brown, and I'm an MD PhD student at University of Louisville and Mayo Clinic, and I'm moderating today's talk. The goal of symposium is to celebrate and showcase the hard work of our Polygent scholars, all of whom have tirelessly worked on their projects over the past couple of months. We have 4 student presenters today presenting on a variety of topics. A fellow Polygent's mentor will be scoring each student's presentation, which will help us determine the prize winners after the event. Please type your questions in the chat if you have any for each speaker. We will allot 2 minutes for questions after each presentation. So today's speakers are Ella Shah, Peter Wang, Taara Shastri, and Aitan Law. So Ella Shah is up first. So you can start sharing now.
Okay, I just quickly want to make sure everybody can see that all right. Okay, great. So my presentation is on cancer anti-angiogenic therapy and mechanisms of resistance. Okay, so first I'm going to be answering the question, what is cancer? All cells have genetic information encoded in DNA. A region of DNA that carries instructions to make a protein is known as a gene. Damage to cells or random mistakes in cell division can cause changes in DNA known as mutations. Mutations that occur in genes can alter the structure and function of proteins. And so cancer arises due to multiple changes in a cell's DNA that alter the function of its genes. Different carcinogens, including tobacco, cigarette smoke, or UV radiation, can cause mutations, thus increasing the risk of developing cancer. The combined action of many genes allows cells to divide. Cell division is regulated by signals from the environment outside the cell and the state of the cell itself. Before dividing, the cell checks for the presence of DNA damage, stress, or nutrient deficiencies that could cause difficulties during division. Many of the genes that are responsible for making cells divide are considered proto-oncogenes, or genes that can promote cancer. Mutations in these genes lead to the creation of oncogenes, which allow cancer cells to divide even without the proper signals or in the presence of cellular stress. Another group of genes called tumor suppressors function in healthy cells to slow down cell division, repair DNA mistakes, and tell cells when to die. Humans usually have 2 copies of these genes because they inherit one from each parent. When both copies are mutated, this may cause cells to become transformed into cancer cells and grow out of control. Now I'm going to be talking about angiogenesis and its role in cancer. An important hallmark of cancer studied extensively is angiogenesis, or the formation of new blood cells from the existing vasculature or blood vessels. Angiogenesis plays a crucial role in cancer growth because tumors need a significant blood supply to provide the nutrients and oxygen required to grow beyond a few millimeters in size. However, expanded blood supply is also a liability for cancer because it allows targeted therapies and chemotherapy drugs to better access tumors. In the early stages of tumor progression, the vasculature supplies enough oxygen and nutrients for the tumors to grow. Cancer cells often consume a large amount of oxygen, take up excess nutrients, and accumulate metabolic waste products. Because of their rapid growth, cancer cells can outgrow the existing vasculature. This causes the tumor microenvironment to become depleted of oxygen, hypoxic, and nutrient depleted, which are conditions unsuitable for cellular growth. However, when tumors become hypoxic, the protein hypoxia inducible factor 1, or HIF1, is activated. HIF1 is a transcription factor that transactivates genes that promote angiogenesis, such as the vascular endothelial growth factor, or VEGF. Then the tumor releases pro-angiogenic growth factors, including VEGF. VEGF is secreted into the extracellular space, binds to VEGF receptors on endothelial cells, releasing proteases and degrading the basement membrane, which causes the membrane to become leaky. Then these endothelial cells, especially the leading cells at the tips of vascular sprouts, known as tip cells, lead the way to the hypoxic environment. This promotes the formation of a new blood vessel that supplies oxygen, glucose, and many other nutrients to the tumor. In essence, the tumor tricks an existing blood vessel into forming new vessels that extend into the tumor environment, allowing the tumor to survive and grow. Anti-angiogenic therapies. Anti-angiogenic therapies prevent tumors from driving angiogenesis. One type of anti-angiogenic therapy uses monoclonal antibodies to target VEGF, which is regarded as a central regulator of tumor angiogenesis. Antibodies are proteins produced by B cells in response to particular antigens. They are created by the immune system to fight infection, but scientists have borrowed their structure to use as scientific tools or medical therapies. Antibodies only recognize a single target or epitope and have two identical binding sites. Monoclonal antibodies are immunoglobulins that can be used to target cancer and other diseases by binding to specific cancer-associated molecules. Scientists can generate many identical copies of a monoclonal antibody that can bind to a specific molecular target. These monoclonal antibodies can inhibit the function of molecules required for cancer growth, flag cancer cells for destruction by the body's immune system, or deliver harmful substances to the cancer cells. A monoclonal antibody called bevacizumab binds to VEGF and inhibits the binding of VEGF to its surface cell receptors. This blocks VEGF-mediated signaling pathways and prevents angiogenesis from occurring. This treatment has proven clinically effective in treating a variety of cancer types. However, it has frequent complications because it inhibits VEGF signaling in normal endothelial cells as well. As a result, treatment is known to cause severe side effects. Now I'm going to talk about mechanisms of therapy resistance. Although anti-angiogenic therapies can prolong a cancer patient's survival, they have a limited impact on the overall survival because cancer cells often require resistance to therapy through numerous mechanisms. Angiogenic tumors can adapt to the presence of angiogenesis inhibitors and acquire the means to continue to grow. The tumors find alternative ways to sustain growth despite the specific therapeutic drug continuing to inhibit its target. The current evidence suggests that 4 adaptive mechanisms allow tumors to resist anti-angiogenic therapies. The first one is the activation or upregulation of different pro-angiogenic signaling pathways within the tumor. The second is the recruitment of bone marrow-deprived pro-angiogenic cells. Both of these mechanisms can eliminate the necessity of VEGF signaling. The third mechanism is the increased parasite coverage of the tumor vasculature, which supports the integrity of the tumor and reduces the need for VEGF-mediated survival signaling. The fourth is the enhancement and activation of invasion or metastasis to supply access to normal tissue vasculature without obligate neovascularization. Now I'm going to talk about a novel treatment. So the main disadvantage of our current anti-angiogenic therapies is that cancer cells often become resistant to therapies by engaging pathways parallel to VEGF. This allows tumors to continue growing and metastasize. A potential novel therapeutic strategy for treating cancer resistant to anti-angiogenic therapy involves blocking the activation of HIF-1 to prevent angiogenesis. Cancer cells are less likely to become resistant to this therapy because the combined action of angiogenesis inhibitors and HIF-1 inhibitors may prevent the use of parallel pathways to continue performing angiogenesis. This therapy could be used on its own or as an adjuvant therapy with treatments including sorafenib or bevacizumab. An efficient way to test anti-HIF treatments on tumors would be to use mouse models of cancer. The treatment could be injected into the mice's tumor, and the researcher could compare certain biomarkers or tumor growth and see if there's any change. Okay, this is my bibliography. Thank you for listening.
Great job. So I guess the question is, how do you hope to use this in the future, or what do you want to do next?
I would hope to look into or potentially like work on creating like methods to test anti-HIF inhibitors, because like they're kind of really new, and so there's not much research that has been done on them, but they could help when paired with anti-angiogenic inhibitors, like reduce, help treat cancer in different patients.
Great. Do we have any more questions? If not, we can move on. Next, we will have Peter Wang.
Okay, hello. I will share screen. Can you guys see the slides? Let's start. So my research paper was on the relationship between COVID-19 vaccinations and death rates. Okay, so my abstract of my paper is, I'm basically exploring the effect of vaccinations stratified by age groups within the United States. So I'll be looking at some data sets on vaccination rates and then compare those to data sets on death rates for different age groups and see whether the vaccination rates of a specific age group will be correlated to the death rates of that specific age group. And what I found was that vaccination doesn't appear to have damped the occurrence of new deaths. So this is supposed to say new deaths, not new cases. Death rates, however, have fallen for more older, highly vaccinated age groups, which would be expected if vaccination reduces the chance of death. Additionally, there's been a question of whether vaccination rates for younger people is important to prevent death rates in older groups. And I found that there doesn't seem to be much correlation between the vaccinations of any given age group and the death rates of other age groups. Okay, so let's look at some historical overview and what's research has been done on this area. So just a little background, within the United States, COVID-19 cases are currently spiking for about the second time. Deaths have spiked for about the third time. And current literature has examined the effectiveness of the vaccine through published case studies. For example, the CDC looked at those admitted to 24 hospitals for COVID-19. They looked at an older age group, I believe it was 65 plus, and they found that vaccination rates were effective at preventing death. So while there have been a lot of different case studies examining different hospitals, there hasn't been much analysis on the effectiveness of the vaccination on death rates as a whole. So my paper tries to add to current literature by looking at comprehensive data for the whole United States and examining the relationship between vaccination and death rates. My definitions of full vaccination are 2 doses of the Pfizer and BioNTech or Moderna coronavirus vaccine, or a single dose of the Johnson & Johnson vaccine. In my paper, I'm just examining the relationship between full vaccination and death rates. So I'm not counting people who have, say, a single dose of the Pfizer or Moderna vaccine. And death from COVID-19 is defined as death where COVID-19 appears as a cause and contributor on your death certificate. And this is according to CDC guidelines. Basically, if, for example, your COVID is listed as the cause of death, or if it's listed as a contributor of death, then that's counted as a COVID-19 death. Okay, so I'll present what I found from my data. So first, I just looked at some vaccination trends as a whole. And my data on vaccination came from the CDC dataset. And this was updated March 10th. So my data ran from about December 14th of 2020 to March 10th of 2022. And you see here that the oldest age groups tend to have higher vaccination rates. The 75 plus age group reached almost 90% full vaccination within the United States. The younger age groups tend to have less vaccination, and the youngest group only reached about 30% vaccination by the end of March of 2020. And then I just did some plotting for death rates by itself. My death rates data came from the United States data.gov dataset, which is managed by the US Department of Health and Human Services. And this dataset was updated, I believe, March 1st of 2022, so also very recent. And it covered death rates from January 1st of 2020 to March 1st of 2022. And in this data set, we're covering new deaths. So just to be clear, this isn't the cumulative deaths. For each date, we're covering the new deaths for each time frame between different months. And we see about 3 spikes for death rates. And again, the oldest age groups tend to have the highest death rates. The pink line here for the oldest age group, 85 and plus, reached about 30K on a second spike, for example. And then what I really needed from this data set was the percentage death rates for each age group. I didn't really want to compare the absolute number of deaths to vaccinations because my vaccination data was about the percentage vaccination rate. So basically what I did for each age group was I divided the number of new deaths by the population of that age group to get the percentage deaths, the percentage rate of new deaths for each age group. And again, we see here that the oldest age group has the highest percentage death rate, about twice as high for a given time as the second highest age group, percentage-wise. Okay, and then I did some analysis on correlation. So I'm first, something I had to do for my data sets, I had to condense some age groups because the age group splits didn't really line up for my different age groups. For example, the data set for death rates may have 5 to 15, age 15 to 20, and then 25 to 40, for example. And my second data set may have the age group splits 10 to 20, 20 to 35, and 35 to 45, for example. So I had to condense some age groups to get proper matching age group splits. And I essentially had 3 age groups, 0 to 24, 24 to 75, and 75 and older. So I first analyzed the correlation for 75 plus age groups. And here we see here I plotted vaccination and death rates for the oldest age groups on the 2-axis graph. So the percentage death is on the left and the percentage vaccination is on the right. And I used the stats model API in Python, and I found a very good moderate negative correlation between deaths and vaccination rates for the oldest age group. We see that deaths spiked for the second time in about January. After vaccinations kicked in, death rates fell a lot, and the third spike was pretty low compared to the second spike. Okay. And then we had the 25 to 74 correlation. I essentially did the same thing. I used the stats model API, and we see that correlation was almost non-existent. We had a second peak here after vaccinations kicked in. The third peak was still about as high as the second peak. So there's very little correlation for our middle-aged group. And then correlation was a little positive for the youngest age group. However, this isn't significant, really, 0.25 for correlation. And then tried Granger causality, which is basically a statistical test for determining whether one series can forecast another. However, my data wasn't stationary, so I couldn't apply the test. So unfortunately, I wasn't able to make any inferences about causality. And then, so our conclusions from correlation is that the high negative correlation for the oldest age group is what we would expect if vaccination rates were effective. However, my research doesn't really show causation. It just lines up with what would be expected if vaccination was effective for oldest age groups. Okay. Then I analyzed the correlation between the vaccination rates of one group to the death rates of other groups. And we see here that the vaccination rates for the oldest age group doesn't seem to really affect the death rates of the middle age group or the youngest age group. And then I did that for the middle age group, aged 25 to 74. And again, there's very little correlation between the vaccination of the middle age group to the death rates of either the oldest age group or the youngest age group. It's all lower than 0.4. So the conclusions for my research overall is that there doesn't seem to be much evidence supporting how vaccinations for any age group necessarily affect the death rates for other group. And we haven't seen really good correlation at all for any group except for the oldest age group. However, this doesn't really mean that vaccinations aren't effective, as there are a lot of other factors at play. That's it. Thank you all for listening.
Do we have any questions? So I guess my question for you is, how would you apply this model to a different problem?
I think another step I guess I could take for this project is maybe comparing to infection rates, and I could apply vaccinations to that as well. Causality didn't really work, but I guess I could try to take the differences of the vaccinations between different time periods. Maybe the differences would be stationary, and if it was, I probably could apply greater causality to the differencing.
Okay, great. Good job. Next up, we have Taara. So you can get started when you feel ready.
Okay. Okay. Are you able to see it? Okay, so my project explores the impact of climate change on hurricanes. Specifically, I analyzed Hurricane Harvey, which took place primarily in Houston, Texas, from August 17th to September 3rd, 2017. So for some introduction, climate change in general constitutes with ocean temperatures rising, air temperatures rising, melting glaciers, and climate change can affect, it can contribute to changing weather patterns, like heat waves and rainfall, increases the longevity of droughts, and increases the severity of natural disasters. For Hurricane Harvey specifically, it made landfall in Houston, or in San Jose, Texas, and caused 68 direct fatalities and 39 indirect fatalities, and about $125 billion of damage. So for some definitions before jumping into my hypothesis and research questions, looking at this first picture, the greenhouse effect, as many probably know, it's a process where gases in the atmosphere like carbon dioxide and methane trap heat from the sun, making the earth warmer. Intensity is the highest sustained wind speed of a hurricane, grouped into 5 categories based on a Saffir-Simpson scale, just pictured here. So Hurricane Harvey is category 4, and it was 130, which falls into the range 130 to 156 miles per hour. Rapid intensification is when a tropical storm's maximum wind speed increases by at least 35 miles per hour in a 24-hour time period. So for my hypothesis, because of my prior knowledge and interest in climate change, I wanted to explore how climate change could affect certain aspects of hurricanes, like total rainfall, rainfall return period, rainfall pattern, and wind speed. I first came up with 4 general research questions that guided my project. Number 1 is, what factors caused Hurricane Harvey to have heavy rainfall and precipitation? Number 2, how did climate change affect the severity of Hurricane Harvey? Number 3, how does the greenhouse effect play into precipitation? And finally, were population dense areas or areas with certain infrastructure more susceptible to flooding? So using articles and other online resources by just looking it up online, I found some key facts about Hurricane Harvey's rain and wind speed. So for the total rainfall, the total accumulated rainfall over the storm was 60.58 inches, or 1,538.7 millimeters. To put this into perspective, if you had an empty outdoor tub, it would have about 5 feet of water in just 4 days. At landfall, the wind speed was 85 miles per hour, or about 137 kilometers per hour. The return period, which is the average time between natural disaster events to occur, is less than 2,000 years. But rare events like Hurricane Harvey make it hard to assign a probabilistic estimate. So looking deeper into the climate aspects, I researched correlations between hurricanes and sea level, ocean temperatures, and even factored in population. I found that generally, Sea level rise causes storm surge flooding to be more devastating and intense. As ocean temperatures rise, there may be fewer hurricanes, but they'll be much more powerful. In the case of Harvey, the warmer ocean temperatures in the Gulf of Mexico made record rainfall and intensity more likely. Regarding population, Houston, especially Harris County, experienced a large increase in minority groups and immigrants prior to Hurricane Harvey. This population shift, along with the geographical area of Harris County, such as more paved roads, caused this area to be prone to flooding and created economic impacts that will be likely impossibly to fully rebuild. So for the procedure, along with online research, I also used Python coding to create contour plots to display the full nature of the storm. In order to do so, I extracted data specifically on Hurricane Harvey from the Goddard Earth Sciences Data and Information Service Center. And then I coded these HDF5 files with PyCharm and Python. So this contour plot, which was the main figure that I created in Python, shows the total rainfall Houston received in millimeters from August 25th to August 27th. So I didn't look at it throughout the entire course of the storm, just a specific few days. So the x-axis is the degrees longitude spanning from -92 to -99 degrees. The y-axis is the latitude spanning from 28 to 31.5 degrees. This scale on the right is the amount of rainfall measured, and I made it go from 0 to 410.2 millimeters. This meter determines the colors that show up on the plot, but the max value of total rainfall based on the code was 1,308.3 millimeters. That is why there's some white overflow in a few areas. So looking farther into the plot, two of Houston's 5 densest areas based on population per square mile, Gulfton and Westwood, which are here marked with red X's, are shown to have gotten little to no precipitation, but the yellow and white areas marked with blue X's, the Gulf of Mexico and Louisiana, received the greatest amount of total accumulated rainfall. So some more results to highlight specific characteristics of hurricanes. I used MIT's single column generator, or SCM for short, in which I changed certain parameters one at a time and kept others constant to show the relationship with precipitation and evaporation. So in figure 3 is an example of what's outputted from the generator. This one had specific parameters of 125 days for time, a surface albedo of 0.1, a wind speed of 40 meters per second, an initial SST or sea surface temperature of 35 degrees Celsius, and a CO2 concentration of 360 parts per million, or ppm. So in figure 2, I took specific data points from the SCM after measuring certain parameters and plotted them using Excel graphs. So precipitations on the y-axis for all of them, and sea surface temperature in graph A, surface albedo in graph B, and surface wind speed in graph C are on the x-axis. So for some conclusions from these graphs, figure 2A shows the direct relationship between precipitation and SST. When the SST gets warmer due to climate change, an increase of atmospheric water vapor fuels hurricanes and increases the chances of heavy precipitation. Figure 2B shows the inverse relationship with the negative slope of surface albedo and precipitation. Surface albedo is the percentage of sunlight reflected by the surface of the Earth. And when it increases, the Earth stores less energy because more of sunlight entering the atmosphere is reflected, which results in cooler temperatures and colder oceans. On the other hand, a warmer planet melts the snow in mountains and glaciers, which results in a lower surface albedo because water is not reflective compared to ice. This decreasing surface albedo in turn causes SST and precipitation to rise. For figure 2C, there's a direct relationship between precipitation and surface wind speed. As the wind near the Earth's surface increases, more friction is created and causes the ocean to release more energy to fuel hurricanes. So this doesn't necessarily cause hurricanes to occur more, but when they do occur, the intensity will be much higher. So finally, other conclusions that weren't displayed through like the contour plot or Excel graphs are relating to the greenhouse effect. Greater carbon dioxide and greenhouse concentrations released into the atmosphere increase temperature and humidity, which then causes more precipitation. So overall, the growing warmer climate results in an increase of precipitation and higher wind speed in hurricanes, which is demonstrated through Hurricane Harvey. And here are some of my references. Thank you.
Great presentation. How do you hope to use this in the future?
Well, for me, it was when I was doing the project, it was just good research because climate change is, a really it's a global problem that's getting worse. And I guess in general, our generation kind of has to deal with it when we get older. And so I want to be able to research more in the future and kind of come up with ways to not fully prevent the effects, but make it at least a little bit better. And I hope to go into environmental science in the future. And my school doesn't have an environmental class, so I kind of have to do my own research. I think this was a good start to looking at Hurricane Harvey and other natural disasters. It's a good start to kind of tie into what I want to do in the future.
Fantastic. So finally, we have Aiton, so you can get started.
Can you see it? All right. So my name is Aiton Law, and I investigated what are the positive and negative regulators of the HPA axis. So the first thing is, what is the HPA axis? Well, its official name is the hypothalamic pituitary adrenal axis, which is really just the three different parts of the axis, which is the hypothalamus, the anterior pituitary, and the adrenal glands. This axis fires any response to stress, whether that be physical or emotional, and it is most responsible for the ever so famous fight or flight response that we have whenever we come into contact with any danger. The first response from the axis is that the hypothalamus releases corticotropin releasing hormone, or CRH. This CRH then travels to the anterior pituitary, which incites the cleaving of POMC into ACTH. This ACTH is then released into the bloodstream and travels all the way down to the adrenal glands, which are on top of the kidneys. And this ACTH releases cortisol, which is the ultimate end product of the HPA axis. And cortisol increases blood pressure, insulin resistance, and gluconeogenesis.
I can't hear you currently.
Hello?
Yep, can hear you
Okay.
now.
When did you stop hearing me? Sorry.
I would just start over at the top of this slide.
Okay, so the inner workings of the axis are ACTH. While it was incited by the release of CRH, it inhibits further release of CRH. And just like how ACTH incites the release of cortisol from the adrenal glands, cortisol inhibits the further release of ACTH. This is all the body's way of making sure that we do not produce too much of a certain hormone, because that could release to negative health effects, and too much of a response can always be a bad thing. So one of the first hormones that I investigated was FK506, or the tacrolimus hormone. This hormone is one of the positive regulators of the axis because it affects the binding site of the glucocorticoid receptor to cortisol. The glucocorticoid receptor usually inhibits the release of cortisol from the adrenal glands, but because FK506 alters the binding site, cortisol can't attach to it, and thus the releasing of cortisol can keep on going. This demonstrates how tacrolimus hormone is a positive regulator of the axis because it incites one of the hormones in the axis to keep on being synthesized. Another hormone or hormones that I investigated was ghrelin and leptin. Now, ghrelin and leptin are hormones that is associated with eating. And ghrelin tells your body that you are hungry, while leptin tells your body that you are full. This is something that's called antagonistic hormones because one does one thing and the other hormone does the exact opposite. And just like how they're antagonistic in respect to how you feel when you're hungry, they're antagonistic in respect to the HPA axis. Ghrelin stimulates the anterior pituitary to release more ACTH in response to hungry or acute stress events, and leptin dampens the HPA axis's response to stress by binding to leptin receptors in the paraventricular nucleus of the hypothalamus of the pituitary. Two more hormones that I investigated are epinephrine and norepinephrine. These are two very similar neurotransmitters and hormones that are found throughout the body, and they both interact with alpha and beta receptors throughout the body. Both epinephrine and norepinephrine interact with the axis in a sort of mixed way. They're both positive and negative regulators of the axis. So when blocking norepinephrine in experimental animals, the animals demonstrated a corticosterone surge. Corticosterone is the cortisol equivalent in test rodents. So by blocking norepinephrine in those rodents, it shows that it is a negative inhibitor because it usually stops the release of cortisol. Whereas when administering both epinephrine and norepinephrine, it is shown to stimulate increased ACTH secretion. So when administering more epinephrine and norepinephrine, ACTH production increases, which shows that it is a positive regulator because the level of which ACTH would normally be is lower than when epinephrine and norepinephrine is present. Further research is required to better characterize epinephrine's and norepinephrine's effect on the HPA axis because their interaction with the axis is still not fully known. I believe we all understand what serotonin is. It provides a distinct reason in the brain that is associated with emotions. Serotonin is released when the body experiences emotions of happiness, calm, and focus. And serotonin has a very well-established positive effect on the HPA axis. In test subjects, when administering any type of serotonin, ACTH levels increase, which means that serotonin interacts with the anterior pituitary portion of the HPA axis because the anterior pituitary is responsible for releasing ACTH. Another emotional hormone that affects the axis is dopamine. Dopamine is just like serotonin in that it is released when we experience distinct emotions such as motivation, productivity, and feelings of being rewarded. But dopamine's empirical effect on the HPA axis is not well solidified. In some patients, it is positively affecting the axis by binding to D1 and D2 receptors and exerts a stimulatory role in the activation of HPA axis in adult rats. But in other trials, like in human trials, when dopamine is administered, it decreases cortisol secretion in about 30% of Cushing patients when undergoing prolonged dopaminergic agonist treatment, which shows how it has both a positive correlation in rats, but a negative correlation in humans. So in conclusion, there are several hormones that can interact with the HPA axis, and there are even more than what I just researched. And we can use these different hormones in order to incite or stop the HPA axis's firing in response to stress.
Great. Good job. So kind of the same question I've been asking other people, but what do you want to do in the future with this?
I would like to conduct some sort of research to try and characterize norepinephrine and epinephrine's as well as dopamine's effect on cortisol or ACTH or CRH secretion, because I believe in respect to like people with Cushing's disease, it would be really beneficial for people like that, because too much cortisol in the body leads to a weakened immune system and lessened bone formation. So if we can figure out whether they have a positive or negative association, we could find a better way to treat their disease.
Great. Does anyone have any final questions for the group as a whole? Okay. Well, you did a really great job, guys. You should be really proud of your work. And congratulations on finishing off these projects.
Thank you.
Thank you.